Chapter 14 is dealing with RNA molecules and RNA processing. What you're looking at here is the family of Tsar Nicholas, which is the royal family in Russia. And what is important about this family, or what ties this family to this chapter, is that the son here, Nicholas, was found to, as a child, have bleeding issues, which we now know as hemophilia. Something as simple as him falling, scraping his knee, or any type of surgery would result in excessive bleeding. And it was determined that he had hemophilia. Now this becomes important and is tied back to what we're talking about, because there are two specific genes or proteins involved with hemophilia that I want to point out. And that is the coagulant factor 8 and the coagulant factor 9. Now, the reason or the importance for these two factors is they are very similar to each other. However, what causes them to differ are introns, and this is through RNA processing. Okay, so you can have proteins that are very close or similar to each other, but they differ based off of their intron regions. And so if you were to sequence the genes, they would look very similar. Okay, and so this led to our understanding of this, led to further exploration of how you can have families of proteins, and they may only differ by the removal of one exon or the rearrangement of these proteins post transcription. Okay, and this was found when they were able to retrieve their bones years and years later, and they were able to do DNA sequencing. They were able to identify this difference. So let's talk about genes, right? We just learned in chapter 13 about transcription and how all DNA is transcribed into RNA, okay? The next step in the central dogma would be understanding how all the RNA that is from transcription is then translated into protein. However, there are some things that we have to keep in mind, and that is eukaryotic cells differ from prokaryotic cells in the concept of colinearity. Okay, prokaryotic cells, i.e. bacteria, tend to be more follow along the lines of the concept of colinearity as opposed to eukaryotic cells. Okay, what is colinearity referring to? Colinearity refers to the direct correspondence or relationship between the nucleotide sequence in DNA and the amino acids of a protein. Okay, that we do not see in eukaryotic cells. There actually is more DNA in a eukaryotic cell than the amount of corresponding amino acids in protein. So this suggests that there is some information that is lost or removed in this process of the central dogma. Okay, so we're going to talk about how genes are organized and how these concept of colinearity and non-colinearity contribute to that. Also looking at the number of nucleotides in the gene, one would think should be proportional to the number of amino acids, as we just previously mentioned, but that is not the case with eukaryotic cells. Okay, actually, if you were to linearize the DNA and then compare it to the linearized mRNA, you'll see that they are not the same length. Okay, actually the DNA is going to be much longer than the mRNA. Right, and you can see this demonstrated through hybridization. We talked about in last chapter, hybridization of DNA and RNA is an indication of active transcription. And so if you were to hybridize mRNA with DNA, you're going to see that there are regions where there will not be hybridization because there are missing portions of the DNA that are not corresponding to or correlating with the mRNA. So with that being said, again, you're going to see more colinearity behavior or colinearity present in bacterial genes, but not as often in eukaryotic genes. And this is just a diagram to hopefully show you this concept of colinearity, which is really looking at the continuous sequence of nucleotides in DNA in respect to protein. So here you have your DNA that is used as a template for transcription to make RNA, which is then made into what we call mature or mRNA, messenger RNA. And then that messenger RNA, every 3 bases is a codon, corresponds to the amino acids that are involved in the primary structure of a protein. Okay, so this would be continuous coding for DNA to RNA to protein. Okay, but again, we do not always see this in eukaryotic cells. This is a more prominent concept seen in bacterial cells. So to investigate this, the question for this experiment was, is coding sequence in the gene always continuous? Okay, and so to investigate this, you mix DNA with complementary RNA and heat to separate the DNA strands, and then you cool the mixture to look at the complementary sequence pairs. Ultimately, this is mimicking some PCR, right? So when you heat the structure, it causes the strands to separate. Okay, that's called denaturation. And then when you cool, you allow for reannealing or renaturation of the sequences. And so when you cool, it should naturally gravitate towards complementary sequences. And so what you'll find is, if there is a fragment of the DNA that is complement to that of RNA, you should have hybridization, as you see here. Okay, but notice there, if hybridization occurs between a DNA strand, as you see here in blue, and an RNA strand, there's going to be portions that loop out or do not bind in a complementary fashion, and that is due to non-coding regions of the DNA that do not have a presence or complementation in the RNA. This suggests that these loops that you see are removed from the RNA. Okay, so that leads us into this concept of introns. Okay, many introns are seen in eukaryotic cells. Okay, when you look at genes or gene structure, there'll be portions of a gene that have exons, which are the portions of the gene that have a coding, and introns that do not have a coding. Both are going to be transcribed into RNA, but introns will later be removed and processed by a concept we call RNA processing. Okay, based off of the cell type in the gene, the number of introns can vary from one gene to the next. Some may have more introns than others, but you're going to see introns more commonly in eukaryotic genes as opposed to bacterial genes. You may see some that are uncommon, but for the most part, you can associate introns with eukaryotic cells. So you'll see here, you have the albumin gene, okay, and cytochrome B gene. The darker color is the exons, the lighter color is the introns. And pay attention to actually the sequence size. The regions of the exons are actually much smaller than that of the introns. Okay, so this should also echo the vast amount of DNA that eukaryotic cells tend to have are more non-coding than coding. Only a fraction of the genes of an intron, a eukaryotic cell, actually code for protein. Okay, only a fraction of the genes, the DNA in a eukaryotic cell code for protein. Most of this is non-coding DNA, and you can see that here. Once these intron regions are removed and the exons, the green regions, are ligated together, meaning joined together, the mRNA size length is significantly smaller than the actual DNA. Okay, so the DNA is transcribed into RNA, and then the introns are removed by RNA splicing. So there are different types of introns. You have group 1, group 2, nuclear pre-RNA, and tRNA. Okay, and these introns, their location are dependent upon the specific type of cell you're talking about, and they have a different splicing mechanism. Some are self-splicing, and then some require what we call a spliceosome, which is a complex of proteins that help genes that help to carry out the removal of these intron regions. You'll see, for example, in group 2 here, they are present in some genes of the mitochondria, the chloroplast, and archaea, and they are, again, self-splicing. Nuclear pre-RNA are the types of introns that are best studied, and they include introns that are located in protein encoding regions or genes that are removed, and the splicing mechanism and how these intron regions are removed are not fully clear, but they know that it requires a spliceosome, so an active cohort of proteins and other RNA molecules called snares to help remove or cut out the intron regions. And then you have the transfer RNA introns. And these are found in tRNA genes in bacteria, archaea, and eukaryotes. And they use another type of mechanism that is more enzymatic to actually cut and release. All right, so the concept of a gene is where you have a gene includes all DNA sequences that encode all of the exons and introns, okay? And these sequences at the beginning and at the end of the RNA are not translated into protein, but they include the entire transcription unit. And the transcription unit, remember, is the promoter region, the RNA coding sequence, and the terminator region, okay? So keep in mind that this concept includes DNA sequences that encode ribosomal RNA, tRNA, and other types of non-messenger RNA. So let's go ahead and look at the structure of mRNA or messenger RNA, okay? Sometimes you'll hear it referred to as mature RNA, okay? And it contains a 5' untranslated region. Or you'll also hear it as a 5' UTR or a leader sequence. This sequence does not code for protein. That's why it's untranslated, right? But it more so plays a role in the initiation process of translation. One of the sequences there is the Shine-Delgarno sequence, okay? And this is important. And then you also have the protein coding region that follows. And then you have the 3' untranslated region, okay? So you have the Shine-Delgarno sequence that is a part of the 5' UTR. Then you have the the protein coding region. And then you have the 3' untranslated region. The 5' and the 3' untranslated regions don't code for amino acids of a protein, but they contain important essential information to start and stop the process of translation, but also for stability of RNA. I won't go through this in detail, but I want to employ you to read through this, as this may be helpful in a upcoming discussion question that you may have to help you understand experimentation and understanding how ribosomes work. So looking back to this structure of a mature RNA, there's 3 primary regions of mature mRNA. There's your 5' untranslated region that I was talking about, and notice in that 5' untranslated region, you have this yellow region here, which is the Shine-Delgarno sequence. This is only in prokaryotic cells. Then you have the start codon, +1 site. Then you have the protein coding region, the stop codon, and then the untranslated region. Okay. There are some differences with eukaryotic cells that we'll talk about. The pre-mRNA processing, okay, specifically talking about eukaryotic cells, is you have the addition of the 5' cap. Now this is unique to eukaryotic cells, and this 5' cap, which is inclusive of a nucleotide that has a 7-methylguanine, so an extra guanine that is methylated at the 5' end, and it's linked by a 5-to-5 bond. This cap is extremely important in the process, stability in the processing of the translation. You also have the poly-A tail. The poly-A tail is added during this processing component, and it contains or consists of about 50 to 200 adenines or bases at the end, okay, following the 3' end of the mRNA. So these are 2 factors that are a part of the processing of RNA. Just to kind of give you another tool that you can use is this table 14.2, which talks about what we call post-transcriptional modifications of eukaryotic mRNA. Okay, and that is the addition of that 5' cap. What does it do? It helps to facilitate the binding of the ribosomes of mRNA, so it increases the mRNA, and this is important in translation. The 3' end, or the 3' cleavage in the poly-A tail, increases the stability of the mRNA, and it helps to facilitate the ribosomes. Hence, remember I talked about in previous chapters that DNA is more stable than that of RNA because, one, it is double-stranded. So the only way you can get a source of stability in RNA, since it is single-stranded, is through this 5' and 3' cap. Okay, so most eukaryotic cells therefore have that 5' cap, and you can see here up close. Okay, here's your ribose. Here you have the 7-methyl, there's a methyl group added, there's your guanine, and so this addition of this 7-methyl guanine cap on the 5' end adds that level of stability to the mRNA, but it also helps with the process of translation initiation. And that process, and let me say, the process of adding that 5' cap is not as easy. I won't go into severe details about it, but I want you to know that there are several different enzymes that play a part in adding that 5' cap, and that initial step is carried out by RNA polymerase. Okay, so that the addition of that 7-methyl guanine cap is a process, and it is required. It requires enzymes to carry it out, one being, or the initial enzyme being, RNA polymerase. All right, so here we have the 3' poly A tail, and you can see here you have a consensus sequence. We talked about what consensus sequences are. If you need to go back and refresh your memory, please do so. And then downstream from the consensus sequence, you have a cleavage site. This is a sequence that is usually 11 to 30 nucleotides downstream. And this is what the machinery, the enzymes that help with the poly A tail addition, identify as where they need to cleave. And then you have the poly A tail, the strand of adenines that are added to the end. Okay. Again, this is a enzymatic process and protein process, because it requires the addition of other proteins to help with this. And it is also inclusive of, it also helps with the protection, right, to prevent the mRNA from being degraded by exonucleases. Remember, the fate of mRNA is to leave the nucleus and go to the cytoplasm, specifically the rough ER. And so on this I'm going to say voyage, okay, from the nucleus to the cytoplasm, there is an influx of enzymes that degrade RNA. And so there has to be a level of protection that is provided for this RNA so that it does not become easily degraded. And that's what we're seeing here by adding this 3' and 5' cap. So how does the splicing work? We talked about the splicing. One, you need the consensus sequences that help. Some of these consensus sequences are located in different regions of this gene structure. So you have the 5' consensus sequence, which is denoted by this GU, either A or G, and then AGU at the 5' splice site. And the 3' end, there is another consensus sequence that consists of CAGG. And then about 18 to 40 nucleotides upstream from the 3' splice site, you have a branch point adenine. So it's an adenine that is used and identified by the machinery to bind there to help activate this active spliceosome, which is composed of 5 RNA molecules and about 300 proteins, to actually begin the processing of splicing out. Let me visually show you excuse me, what that looks like. So there's your 5' splice site consensus sequence. Then you have your 3' splice site consensus sequence, and going upstream about 18 to 40 nucleotides, you're going to have that branch point adenine. Okay. Notice these consensus sequences are sandwiching the intron region. Okay. So here's your exon. That's the protein coding region. Exon 1, exon 2. Your other protein coding region, this is the non-protein coding region. So you have these consensus sequences that that occur at the beginning and the end of the intron, so that the machinery can identify it and begin to splice it out or cut it out. So that becomes a 2-step process. Okay, so the 5' end of the intron is cleaved, and I actually will use this diagram to explain this. Okay, so you have the 5' end of the intron that is cleaved upstream from the exon, okay, and attached to a branch point adenine, which is going to be about right here. The 3' end of the intron is cleaved from the downstream exon. Okay, we have that 3' splice site. And so once they are, this lariat forms, it binds there, it helps to form a lariat. Okay, that's what you see here. And that lariat is released from the exon 1 and exon 2 from this gene. And once the lariat is removed, which is the intron region, then you have the exon 1 and exon 2 that are joined together. Okay. And this process of joining them together, okay, allows for the formation of the mRNA. Now keep in mind that the exons may not always be joined together. Let's look at it a different way. Here you have U1, excuse me, that will bind to the 5' splice consensus sequence. U2 is going to bind to the branch point adenine. Once these two factors, U1 and U2, bind to and their consensus sequences, you have U4, 5, and 6 that come in and form this complex to help form an active spliceosome. And then once that active spliceosome forms, it helps to form that lariat. U1 and U4 are removed. There is no U3. Okay. Once the lariat is removed from the active spliceosome, then you have the exons that are joined together, and that lariat is released. Okay. Let me go to, I'm going to skip those two, and I want to go to this next concept. I'm going to go on down to editing really quick, okay, because I've kind of gone over this already. With RNA editing, this editing process occurs after the coding sequence has been transcribed, okay, and what plays a major part in this is guide RNAs, okay. So another way of saying that is once you have the transcription of the gene, okay, you have to go back, or the cell has to go back and edit this component because the translated protein has amino acid sequences that differ from the encoded gene. So a guide RNA is going to contain sequences that are partly complementary to those segments of unedited mRNA, and these two molecules will undergo base pairing in these sequences. So they play a role in guiding this process. What you see here is messenger RNA that is produced from mitochondrial genes of a parasite. That parasite is what you see in purple. And it undergoes extensive RNA editing. Okay, and we see this is what's called the African sleeping sickness. So let's look a little bit more into guide RNA. Okay. So the guide mRNA has sequences that are partly, okay, complementary to those of the pre-edited mRNA, and it pairs with it. Let me see, I want to make sure I can point that out, okay. After pairing, okay, so you have this pairing that occurs, okay. After pairing, the mRNA undergoes what we call cleavage, and new nucleotides are added, with the sequences in the guide RNA serving as a template. The ends of the mRNA are then joined together. Okay, so the guide RNA ultimately adds nucleotides to the mRNA that were not encoded by the DNA. Mature eukaryotic RNA is produced when pre-mRNA is transcribed and undergoes several types of processing. Now keep in mind, we've already talked about the splicing component, so where you have all the sequence that is transcribed into RNA, okay, and then you have these lighter regions, which are the introns, that have to be spliced out. So you have the splicing that it occurs, but you also have the 5' and the 3' cap that is added with this the 3' in that strand of A's. Okay, here you have the intron regions that are removed, and then the rejoining or the ligation of these various exons. Again, I want you to keep in mind that even though this diagram is showing you all of the exons regions being joined together, that is not always the case. There are instances where some exons are not ligated or rejoined to form the mRNA. Okay, you can use this diagram here. This is a representation to show you the nucleotide sequence of a gene for a human interleukin 2. And in this, it includes the TATA box, which you see here, the start site, the stop site. It includes introns and exons. So the exons are going to be in your darker green, and the introns are going to be in your lighter green. And it also includes the poly A tail and the 3' cleavage site. Okay. So this is visually showing you what the sequence would look like. You can pull this up on BLAST or on the NCBI website, which has the sequence of all genes present in a eukaryotic cell. So you can pull up any gene, and then you should be able to find, given these consensus sequences, you should be able to find these components within a gene. But another thing that I want you to pay close attention to is the fact that the non-coding introns occupy a larger portion of the gene than the actual coding region, the exons. So this also helps to reaffirm the reason why we have more DNA than we actually have coding of for proteins. So what is the structure of transfer RNA? Because we've talked about mRNA, we've talked about the small nuclear RNA, which helps with the process of RNA processing. The transfer RNA is a rare modified RNA, has rare RNA nucleotide bases, ribosome and pseudouridine. And the structure that you see with a tRNA is a cloverleaf-like structure. It also has an anticodon region or anticodon loop. And I just want to visually show you, these are the two rare RNA nucleotide bases, and they resemble that of uracil. Okay, so it's a modification of uracil to these two bases. Now, this is the structure of a tRNA, and this is just showing you in different ways, but this is how you would more so see it. You have the different bases or arms. This is the acceptor arm. This is where your amino acid is going to be bound or attached, and that amino acid that is bound here should correspond to the anticodon loop here, which is the 3 bases that correspond to this amino acid here. Looking at this transfer RNA, know that transfer RNAs are processed in both bacterial and eukaryotic cells. So there's a processing that has to occur for these. And you will have, in some instances, introns that have to be removed from the tRNA so that it can be a mature tRNA that has no intron regions and has the acceptor arm here, those 3 bases here that correspond to the anticodon region here. All right, ribosomes. Okay, so we've talked about mRNA, we've talked about snRNA, we've talked about tRNA. And again, remember, tRNA does not become protein. snRNA does not become snRNA does not become protein either. These are RNA molecules that do not become protein, but they take on secondary or dimensional shapes or structures that play an important role in the process of transcription or translation, primarily translation. Okay, the structure of the ribosome, you have a small ribosome subunit and a large ribosome subunit. Okay, the number of the subunits, okay, are either 70S for bacteria or 80S for eukaryotic cells. The S stands for sedimentation, or the Svedberg unit, and it's referring to the sedimentation rate, the rate at which it sediments or through centrifugation. Okay, so this is why they don't add up. They're not supposed to or meant to add up. So in the 70S, the large subunit is 50 and the small is 30. In eukaryotic cells, the large subunit is 60, the small is 40. In a part of the ribosome, you have ribosomal RNA that is embedded within the ribosomes along with proteins. Okay, And this is how you have the actual ribosomes that are that play a role in translation. The ribosomal RNA genes differ based off of the different organisms, right? In E. coli, there's about 7 copies of ribosomal RNA per genome. Humans is 280. Frogs have 450. So you can see that the ribosomal genes per genome differ based off of the organism, and it does not always have to do with complexity. All right, so what happens to the ribosomal RNA once it's transcribed? Once the ribosomal RNA is transcribed, it is processed. Okay, so you'll see here in A, this is prokaryotic RNA, and B, it's eukaryotic RNA, and they're produced from a precursor RNA transcript that are methylated. Once it's methylated, it's cleaved, and then it's processed to produce mature ribosomal RNA. Eukaryotic 5S RNA, right here, is transcribed separately from a different gene. Okay, so eukaryotic 5S is transcribed separately. All right, then we are going to jump into RNA interference. Sometimes you may hear it referred to as RNA and then lowercase I. RNA interference limits the invasion of foreign genes and censors the expression of their own genes. Okay, so RNAi is really important in blocking foreign genes from being able to express their own genes. And there are different types of small RNAs, and they are processed differently. Let's look at some. You have microRNAs, and then you have small interfering RNAs, or siRNAs. How they work are different based off of the cell, but ultimately their role is to inhibit translation. And small interfering RNAs, their role is to actually mark the mRNA and then cleave it. Okay, but the whole purpose of these RNAi is to prevent or reduce the invasion of foreign genes and censors the expression of their own genes. In microRNAs, they use a molecule called dicer, and dicer helps to break down or cleave the RNA, and it does that by helping to form this terminal loop, what we call a hairpin. And then it also uses RISC, which is a compound or complex that helps with the silencing. Okay, so you'll also see in instances where microRNAs are shown to silence expression of genes. MicroRNAs have been associated with playing a role in cancer progression, various human diseases. We see them also in plants. So microRNAs are very important, and they are located in the intron regions. Okay, same with siRNA. We actually use siRNA in research for understanding what happens if we were to silence certain genes. What would the effect be? This diagram here is table 14.5, and it's showing you the different types of the differences between siRNA and mRNA and microRNAs. So it's really helpful to look at and understand this table. Know that the size of the siRNA and microRNAs are very similar, but their role is different. You're going to see siRNA more so in inhibiting transcription and chromatin modifications, whereas microRNAs, you're going to see it involved in the degradation of mRNA and inhibition of translation. Okay, so this is really key. It's really important. And then lastly, we have Pee-wee RNAs, or Pee-wee interacting RNAs. And these are primarily found in germ cells of animals, and they inhibit transposons. So their role, these Pee-wee RNAs, are really responsible for inhibiting transposons. Okay. CRISPR RNAs are found in prokaryotic cells, and you, I'm sure, have heard a lot about CRISPR RNA. It was a few years ago really a hot topic in science, as a scientist in China was using CRISPR to cause gene modifications in these twin girls, okay, specifically dealing with HIV/AIDS. And CRISPR, the etiology of CRISPR is prokaryotic or bacterial originated, and it is their form of defense. Like we have our immune system, the CRISPR is their their type of defense. And now scientists have taken that information and are now utilizing it as a potential tool for regulating gene expression by modifying certain genes. As you can see here, CRISPR RNAs are meant to defend prokaryotic cells against the invasion of foreign substances, i.e. bacteriophages and plasmids. Okay, so you can see here you have the different phases that we talked about: the acquisition phase, the expression phase, and the interference phase. Okay, and the components, these different components of the CRISPR, play a role in each of these phases in helping to build memory to foreign DNA and to mark it so that it cannot gain access and incorporate its genetic information into the host cell. And lastly, talk a little bit about briefly about long non-coding RNAs. These long RNA molecules do not encode proteins. Many of their functions, their role function in controlling gene expression. Examples like enhancer RNAs, they're transcribed from enhancers and they play a role in controlling gene expression. And this can be very broad depending on the cell type. Circular non-coding RNAs serve as sometimes decoys for microRNAs, right? But these are ultimately found to help regulate gene expression. So this is chapter 14.